A patient crashing into septic shock is a macroscopic crisis. Blood pressure drops, organs starve, and the vascular system dilates uncontrollably. But this cascading failure begins in milliseconds on the microscopic surface of individual immune cells. The innate immune system operates as a high-speed, immediate response radar network. It scans for molecular patterns instantly, functioning as the body's primary threat detection grid. To maintain this surveillance, the network must answer 3 precise questions. Is a molecule foreign? Is it dangerous? And exactly where is it located? This diagram illustrates the sensing logic. The top left green icons, lipopolysaccharide, flagellin, and double-stranded RNA, are pathogen-associated molecular patterns, or PAMPs, microbial fingerprints. Below them are endogenous stress signals, the orange DAMPs. Molecules like ATP, uric acid, and mitochondrial DNA signal massive cellular injury. To read these 2 threat signatures, the cell relies on a sweeping category of receptors known as pattern recognition receptors, or PRRs. The innate immune system relies on cellular geography. The physical location of these PRR scanners, whether on the outer membrane, inside a cargo bay, or floating in the cytoplasm, dictates the specific systemic alarm the body will sound. The cell's first line of defense guards its outer boundaries. This perimeter is manned by a family of sentries called toll-like receptors, or TLRs. On the top edge of this cell membrane diagram, we find the surface TLRs. These receptors face outward, scanning the extracellular fluid for bacterial threats approaching the cell. TLR2 identifies peptidoglycan on gram-positive bacteria. TLR4 senses LPS on gram-negative bacteria. Once locked onto LPS, TLR4 triggers an internal cascade. Tracing the top row, activation passes through MyD88 and TRIF, down to IRAC, ultimately activating NF-kappa B. This pathway forces the cell to synthesize and release pro-inflammatory cytokines, TNF-alpha, interleukin-1, and interleukin-6. By detecting the threat at the surface membrane, the cell initiates a response restricted to localized extracellular recruitment, assuming the cellular wall has not yet been breached. When a cell phagocytoses, or essentially swallows, a foreign particle, it physically traps that material inside an isolated internal compartment. Focusing on the purple structure on the left side of this diagram, we see a newly formed endosome. Notice the receptors here, TLR3, TLR7, 8, and TLR9, are pointing inward. These endosomal TLRs act as cargo bay scanners, interrogating engulfed material for hidden nucleic acids. Their targets are precise. TLR3 hunts for double-stranded viral RNA, while TLR9 targets unmethylated CpG DNA. Human DNA is heavily methylated to regulate gene expression, whereas bacterial and viral DNA typically lack this modification. Therefore, unmethylated CpG serves as a distinct microbial fingerprint. Once these endosomal receptors confirm the presence of foreign nucleic acids, their signaling output drives heavy interferon production to halt viral replication. Endosomal detection marks an escalation. The threat has breached the outer perimeter, but remains sequestered inside the endosomal membrane, allowing the cell to prepare a targeted antiviral counterattack. The most dangerous scenario occurs when a virus bypasses the endosome entirely, dumping its toxic genetic payload directly into the free cytosol. To counter this, the cell deploys RIG-I-like receptors, or RLRs. As shown in the yellow cytoplasmic region of the diagram, RIG-I and MDA5 function as the cell's internal antivirus software, scanning for free-floating viral RNA. Normal human mRNA is chemically capped. Many viral RNAs possess exposed, uncapped 5' triphosphate ends. RIG-I identifies this specific structural anomaly to recognize the RNA as foreign. MDA5 identifies long double-stranded RNA from large viral replication events. Tracing the yellow row, RIG-I and MDA5 activate MAVs on the mitochondrial membrane, triggering a cascade that drives mass production of type 1 interferons. RNA viruses aren't the only threat in the cytosol. Free-floating DNA in this compartment triggers the C-gas sting pathway. DNA outside the nucleus, whether from a virus, broken mitochondria, or a tumor, activates the enzyme C-gas. This produces the messenger molecule CGAMP, which then binds to the sting receptor on the endoplasmic reticulum. Whether RLRs detect RNA or C-gas detects DNA, The presence of free nucleic acids in the cytosol universally triggers the production of type 1 interferons and the activation of natural killer cells. In addition to monitoring for foreign pathogens, the system must also detect sterile stress and catastrophic internal damage. For extreme cellular distress, the cell relies on nod-like receptors, or NLRs. On the right side of the diagram, NLRP3 monitors for intracellular stress signals. NLRP3 triggers upon specific physical crises: potassium efflux, lysosomal rupture, reactive oxygen species, or severe ATP depletion. Hitting critical mass, they trigger an explosive assembly. NLRP3 recruits ASC, activating caspase-1 to form the inflammasome. Active caspase-1 immediately cleaves an inactive precursor into mature interleukin-1 beta and initiates pyroptosis. Pyroptosis is programmed cellular suicide. The cell swells and violently bursts, destroying its compromised machinery to release an unavoidable warning signal to the immune network. This microscopic molecular surveillance grid directly drives the macroscopic clinical realities encountered in critical care. In sepsis, LPS overstimulates TLR4, while cellular damage triggers NLRP3, generating a lethal cytokine storm. In viral ARDS, viral RNA triggers TLR3, RIG-I, and MDA5, creating an alveola-destroying interferon surge. The system follows consistent logic. The PAMP is the fingerprint, and the PRR is the scanner. The surface and endosomal TLRs function as the physical door and cargo bay sensors, while the RLRs operate as cytoplasmic RNA antivirus software. Within the cell, C-gas identifies displaced DNA, while NLPR3 detects signs of internal physical damage. In the innate immune system, biology is geography. By understanding exactly where a receptor sits and what pattern it binds to, clinicians can precisely track and predict the cascade of systemic immune failure.